transistors that gate currents and voltages. They have valves that gate fluid flow. But the whole idea here is that we've reduced something that's very chaotic, the idea of mixing fluids together and so forth, into a very controlled, computer-controlled process. The end result, though, is strikingly similar. So on the left-hand side of this montage is a typical integrated circuit board. You might find it in your computer at home. And the right-hand side is an integrated DNA sequencer that was developed at Berkeley to integrate all of the processing steps involved in DNA sequencing, and there are quite a few of them, all on the nanoliter scale and with no robots or pipetters or anything like this. Everything is driven around by voltages and pressure. So this is the inspiration that drove me to Jerry's lab, where there's this beautiful paper talking about in vitro evolution and this kind of cyclic process of selection, amplification, mutation, growing up in the tubes, propagating to a new tube. It's this kind of repetitive process, and it seemed kind of ripe for a microfluidic approach to take the human out of the equation and begin to observe evolution in real time. So this is the circuit that we devised in La Jolla. It's our microfluidic mixing circuit. And the idea behind this is just a series of channels. It is a reactor for incubating our molecules, a reaction mix. It also has a series of valves and some reservoirs. So there's an input reservoir where our reagents go into this device, and there is an output reservoir where our reagents are removed and stored for further analysis. There are also valves inside this network of channels. So the channels are actually shown here in bright field, and they're filled with a fluorescent dye. There are 3 two-way valves which mitigate fluid, or which propagate fluid flow around the loop on the outside. And there are also 2 access valves which control access to and from the circuit up here. And so I want to show you here is how, under computer control, we can do this process of serial dilution that I showed you previously, where we're passaging molecules, cells, whatever have you, from tube to tube. But now we're actually doing that process of serial dilution under complete computer control. So the whole dye is initially filled with, or the whole circuit is filled with dye initially. We begin to pump our diluent, which is buffer here, has no dye in it, so you're seeing it disappear on the right side. We've held the two-way valve on the left-hand side closed, and that is actually holding the carryover material that's going to be propagated to the next generation once we've diluted in our next starting reagent. We close off the access valves during mixing and actuate those 3 two-way valves in serial, and that affects microfluidic pumping and mixing. So this is one serial dilution carried out in movie format. So we assembled this circuit into a very larger system that would be capable of monitoring the real-time fluorescence of our sample that is being diluted and amplified in this circuit. We included a fluorogenic dye to be able to track how much RNA was produced. And then we set the computer to task. We set a growth threshold. We set a dilution rate, which is actually set by the circuit geometry, so that every time we want to dilute our population, it dilutes by a factor of 10. And the computer again begins to look for growth of fluorescent signal to monitor the intensity and the growth of new molecules. And so here are 500 transfers under computer control. There are individual places where we had to remove the molecules, remutagenize them. This is a very stringent selective process, and we want to keep the number of individuals participating at a very high level, which necessitated periodic mutagenesis. More importantly was the selective pressure. So our molecules started out as kind of piggies. They needed a lot of that substrate promoter tag, that substrate. They needed a lot of it in order to catalyze the chemistry, to catalyze that ligation chemistry to participate in the amplification process. And so what we hypothesized was that if we basically subjected this population to the pressure of reducing substrate concentrations, that by starving the population, we would encourage the growth of superior enzymes, molecules that had better ligation kinetics and could more efficiently use substrate. And so this is a Michaelis-Menten plot. For our graduate students who are going into qualifying exams, you'll be able to know what this data is. It's a plot of the observed rate of the molecules as a function of the amount of substrate that they were using. And a higher rate means a faster reaction. Our starting molecule needed more substrate in order to ligate faster, whereas our evolved molecule, which was pulled out at the end of this experiment, needed virtually no substrate, having been raised on a diet, on a severe diet. This was caloric restriction before, but in molecules. Very little substrate in order to catalyze the reaction at an equivalent rate. All right, so the story here is, so this was Superman molecule. Unfortunately, the results that came out of this experiment were clonal in the sense that we pulled out multiple individuals at the end of this experiment, but they were all the same molecule. And this is, in hindsight, perhaps no surprise, because the system is so selective that in the end, it's only the master sequence, the best possible sequence that can exist, and all copies of itself that came out of this. And so we began to think, how can we begin to look at sequence space, get this idea, what is the functional density of sequence space in RNA? How many other different kinds of survival strategies can we find? How can we get out of the ocean where everybody is competing against one another and look for more diversity in function and structure? So this is where the idea of parallelization comes in. Parallelization is one of the landmark capabilities of miniaturization, the idea that if you can make one, you can make a bazillion. And parallelization also has overtones in the story of evolution that I'm telling here today, which is that our birds from the Galapagos are essentially this experiment in parallel evolution. They were not all racing against one another on the same island for the same seeds and so forth. They were distributed across this archipelago in the Galapagos. And depending on what resources were available there, different birds flourished. And they flourished in very high diversity with some very interesting characteristics. There's a book actually called Beak of the Finch, which describes this whole setup. Very interesting. So I kind of took inspiration from this, and I thought, well, why don't we just populate a whole bunch of my reactors with different starting molecules and see if we can come up with some very interesting solutions. And the idea is that one island here is one reactor, and we could just take that one reactor and array it over the whole chip. And there's our parallelization. And it turns out that at this point in time, it helps to be a little cleverer than this, because this would be a really tedious device to make. We'd have to make lots of microscopes, lots of valves. It's just a little bit too much. And there are actually answers simpler than this in chemistry, which is emulsions. And everyone's familiar with emulsions, and we'll come back to this later on. Mayonnaise is an emulsion. But these are stable dispersions of water droplets in oil. And these emulsions can essentially function as islands because these droplets do not mix. So if we know how big the droplets are, we can take our initial population of RNA molecules, dilute them down so that on average there's one molecule per droplet. And now there's no race anymore. All you have to do is catalyze the chemistry that we described earlier and replicate in your droplet. There's no competition. And we get to see who are the comers, what are the strategies for survival, not just who's the best. The problem here, though, comes in how we make emulsions. So in the laboratory, we make emulsions in a tube. We make emulsions by taking oil and water and a little bit of surfactant stabilizer, and you take your homogenizer, your blender, and you stick it in the tube and whir away. And that's when you get this over here. And the people in the audience who are biochemists and molecular biologists who work with proteins know that this is a recipe for disaster if you have to work with proteins. So it turns out there's an elegant solution in microfluidics for making stable water droplets in oil, and it's a very simple circuit design. It's shown here on the left-hand side, where oil phase is coming in from the top and the bottom channels, and our aqueous phase is coming in from the left-hand side, and these are literally pumped in by syringe pumps. And now we're no longer cooking. We're not mixing some oil and some stabilizer and putting the blender in. We're actually doing science. We have syringe pumps at known rates that are pumping fluids in. We have a circuit geometry, which is in my AutoCAD file, and I can ship it to any lab in the world, and they can put this system together and make droplets. And we can actually program droplet size. The more oil we have, the more shearing, the smaller the droplets get, and similar for larger. It's actually complete control of parameter space. So no more blending, science. The only problem is that this is one stream, and it's looking at nanoliters per minute. It's only able to generate very small numbers of droplets. And in the game of evolution, it's numbers. It's all about how many can you look at. And we want to look at millions. We want to look at solutions that are 500 microliters, mils of stuff, perhaps even liters if we got really ambitious. And when you're working at nanoliters per minute, a liter is a totally unimaginable amount of stuff that you have to make. That's an ocean. All right, so to get around this problem, or at least to begin to address it, we constructed a parallel array of nozzles using this idea, but in a much more clever way. The idea that